So Babesia like here is just like Plasmodium. So it is also an intraerythrocytic protozoan that causes malaria-like illness in animals, particularly cattle and sheep. But what's intriguing about this parasite is its transmission. So it is transmitted not by mosquitoes, but it is transmitted via ticks. And Babesia doesn't just affect animals. Some species, like your Babesia microti, can infect humans and causing babesiosis. So this is actually a disease resembling malaria, meaning the life cycle mimics your plasmodium. So it is transmitted by Exodus ticks. So these are the same ones that carry the Lyme disease. So we will be talking the Lyme disease on your bacteriology class. Okay, so this is the definitive host of your Babesia. So if the definitive host in your malaria is the mosquitoes or your female Anopheles mosquito, in here the definitive hosts are the ticks, the deer ticks to be specific, the Ixodes capillaris, and then the intermediate host would still be the humans. So this is the life cycle. So the life cycle again mimics the same. So it has almost the same life cycle as your plasmodium. So in humans, the same story. The asexual stages begins when sporozoites enter the red blood cells after being introduced via a tick bite. So inside the RBCs, these sporozoite transforms to become trophozoites, multiplying and causing the symptoms of babesiosis. So, but the story doesn't end there. The sexual stages, right here, the sexual stages unfolds inside the tick. So, in here, just the tick. When a tick feeds on an infected host, it ingests the gametocytes. So these gametocytes are the sexual forms of the parasite. So these gametocyte travels to the tick's intestines. It travels to the tick's intestines where they undergo sexual reproduction and then results to zygotes, migrates to the salivary glands, transforming to become infectious sporozoites and ready to be transmitted to the next host. Okay, so now what I want you to remember is when you examine Babesia on a blood smear, you'll only find trophozoites. You'll only find trophozoites, either as ring forms or the distinctive Maltese cross. So in here, you you could see the difference that this absence of schizons and gametocytes is a key distinguishing feature between babesiosis and malaria during microscopic evaluation. So unlike malaria that you could actually see schizons and gametocytes, in babesiosis you cannot see any schizons. No schizons or gametocytes are actually found in the PBS or during microscopic examination of these certain parasites. So, for the pathogenesis and clinical features, let's start with the mild cases. So, it is actually commonly caused by Babesia microti. Babesia microti. So, symptoms often mimic the flu. So, we have here fatigue, weakness, fever, chills, sweats, headaches, and we have loss of appetite, nausea, and even dry cough. These are actually due to the destruction of the red blood cells. So it has the same. So your parasites also causes the destruction of RBCs and the inflammatory response it triggers. So if there's destruction, we have the immune response to inflammatory response to mediate the destruction. So in severe cases, it is particularly in people or particularly common in people with weakened immune system or specifically with people with no spleen. So these patients can develop severe anemia or acute respiratory distress. So acute respiratory distress, also known as your ARDS. So we have clotting issues like your DIC. DIC stands for disseminated intravascular coagulation. We have heart failures, kidney failures, and even splenic infarcts or ruptures. So it is therefore life-threatening if untreated. So for species like Babesia divergens and Babesia duncani, infections are far more aggressive. So primarily affecting splenectomized or immunocompromised individuals with high mortality rates. And again, what's striking in this clinical features is the absence of schizons and gametocytes in the blood films. So unlike malaria, you could see schizons and gametocytes in your PBS, but for babesiosis, there is no schizons or gametocytes being observed. So diagnosis on babesiosis relies heavily on blood smears and PCRs and serology. So to those patients suffering from asymptomatic babesiosis, the confirmatory test will always be serological and PCR or molecular level. Okay, so let's focus on the diagnostic hallmark of babesiosis. So that would be your PBS. Just like your malaria, the hallmark or the gold standard for diagnosing symptomatic babesiosis would be PBS. So when examining a blood smear, one key feature to identify babesia is this one. This. This is a Maltese cross form. So this is a tetrad structure formed by dividing ring forms. So these are early stages of the trophozoites within the red blood cells. This is a very unique pattern. So this unique pattern is actually a dead giveaway for Babesia infection. So if you see this, especially in Babesia microti, so if you see this Maltese cross form, think of Babesia. In addition to the Maltese cross, you'll also see ring forms similar to those in malaria. But again, the difference between your Babesia and from your malaria is Babesia again lacks the schizons and gametocytes. So in blood films, you can actually distinguish your Babesia and your Plasmodium with this um, difference or with this lacking stages, the schizons and gametocytes. Okay, we have here the treatments. We will not focus more on the treatments, so we're not doctors. So here, let's go back to the case that was presented to you during the lecture session. So this was a case about Alex, a 22-year-old college student who experienced fever after a Palawan adventure. So our first question was, how did Alex get infected in Palawan? So as we discussed earlier, Alex was likely bitten by an infected Anopheles mosquito while in Palawan. So the female Anopheles mosquito is the primary vector for malarial transmission. And in tropical regions like the Palawan, the risk is actually higher than Siargao or wherever um, place here in the Philippines. So question number two, why didn't Mia and Ryan get sick? There are actually several possible reasons for this. So number one, there's no exposure to infected mosquitoes. So it is possible that Mia and Ryan were simply not bitten by mosquitoes carrying the Plasmodium parasite. So even in endemic areas, not everyone gets exposed to the infected mosquitoes, 'di ba? Remember what I told you? If you're being asked of the mode of transmission, you always indicate the bite of an infected female Anopheles mosquito because not all Anopheles mosquitoes are actually infected. Okay. Another reason could be immunity. So, as you go on to this case, you would know that the culprit of this case is Plasmodium vivax. And Plasmodium vivax is known to have a dormant stage in the liver. What stage is that again? It is the hypnozoid stage. So if Mia and Ryan had prior exposure to malaria, they might have developed partial immunity. So this immunity could be due to their body's ability to control the parasite, so preventing it from becoming active or causing illness. So additionally, we have also other factors like the genetic factors, like the presence or absence of the Duffy antigen. You remember that one? Duffy antigen, your FY antigen on the red blood cells. So the Duffy antigens, if you remembered, acts as a receptor for Plasmodium vivax to enter red blood cells. So meaning people who lack the Duffy antigen are resistant to Plasmodium vivax infection. So if Mia or Ryan lacked the Duffy antigen, they might have been immune to the parasite. No? So that would be another reason. Or before we go to question number three, or it could be luck. Sometimes exposure to mosquitoes is a matter of chance. Not everyone gets bitten at the same frequency or time, and it's possible that me and Ryan were simply lucky enough to avoid mosquito bites. So, question number three. Why does malaria causes cyclical fever? So malaria's cyclical fever is caused by the synchronized rupture of infected RBCs. If you remember, your RBCs would would rupture. So as the Plasmodium parasites mature inside the RBCs, remember that they would eventually burst. So they eventually burst out, releasing merozoites. So these process triggers the release of inflammatory cytokines, which lead to fever. So for Plasmodium vivax, in this case, as you could see, the fever seemed to occur every 48 hours. So this cycle typically occurs every 48 hours, which is why Alex experienced fever in waves. So in waves. Next, what do you think is the suspected agent of this case? It's very obvious. So, given Alex's symptoms and the cyclical fever, which is every 48 hours, the suspected agent is Plasmodium vivax. So, this species is very common in tropical regions like Palawan. So, we can we can rule out Vale because Vale is one of the rarest Plasmodium being found here in the Philippines. So if you're being asked to choose the best answer will always be Vivax. It is very common in Palawan and it matches the clinical findings, including the presence of trophozoites and schizons in the PBS. Okay, so I guess this ends my lecture video about malaria and babesia. So I hope you have learned something. These are my references. Aside from these references, always refer to CDC. Okay, thank you very much.